So Dominic's already told you that, and I think you know that mitochondria are the power plants of our cell. They're sort of floating around inside the cell, and what they're doing is basically burning fuel. So we eat sugars, fats, and proteins, and we need oxygen. That's the reason we need oxygen, to burn the fuels effectively to generate energy. And we carry the energy around in this chemical called ATP. So mitochondria are our power plants, or our furnaces where we're burning fuels. And there was a nice paper a few years back that I think sort of puts this in context about, even at rest, the human body has to constantly generate about the same amount of energy as a bright light globe. So we are having to generate about 100 watts of energy, even when we're not doing anything. Well, so to do that, we have to consume about 400 litres of oxygen every day. So oxygen's about 20% of the air. So as we breathe in, we need 400 litres of oxygen to burn the fuel. And we turn over our own body weight in that little energy carrier every day. So we're just making it and using it all the time. About 65 kilograms of ATP as our heart is pumping and our brain's still working. And if you had to go and buy that in a chemist, it would cost you about $3 million a day. So one of the numbers, again, that's kind of relevant is to think that our brain is only about 2% of our body weight, but at rest is using about 20% of the total energy use. And so that's why it's not surprising then that the brain involvement, neurological issues, are one of the most common things in mitochondrial disease. Put that out of reach so I don't go to click it. Dominic, obviously given you some of my slides before. So Dominic's already told you that mitochondria are encoded, if you like. They're these power plants in our cells, and they comprise something of the order of probably about 1,000 to 1,500 different proteins. Most of those are included in our nuclear genes, and the nuclear genes are arranged on 23 pairs of chromosomes. So we get one pair from mum and one pair from dad. There's about 20,000 genes in all that encode everything in our body, and that's in a string of letters. Our total DNA is about 3 billion base pairs long, so 3,000 million base pairs or letters. The mitochondrial DNA is different. It's only really quite small. It's just this one circular chromosome, 37 genes in all, and it's only 16,500 base pairs, so it's a tiny amount of our total DNA. And as Dominic said, the unusual thing about mitochondrial DNA is it's maternally inherited. And basically, in the human egg, there's 200,000 copies of the mitochondrial DNA genome. So half the DNA in the egg is mitochondrial DNA, and that's basically swamps the small amount of mitochondrial DNA in the sperm, and so your mitochondrial DNA is passed down the maternal line. So what that means is that if you were to look at my daughter Emma's maternal line and look at her mitochondrial DNA and compare it to her mother or grandmother, great-great-great-great-great-great-grandmother, the mitochondrial DNA is identical there, unless a new mutation has arisen in a maternal line, whereas with each generation we're only passing on half of those nuclear genes. And again, most people have only heard about mitochondrial DNA either from watching forensic shows, or the other thing that you sometimes hear about is tracking population movements around the world, or tracking the female population movements around the world, if you like. And so if you're a science nerd, you might send off your daughter's DNA to the National Geographic program after she's turned 18, and look at how it matches up. There's a book called The 7 Daughters of Eve, if you're interested in that sort of thing. And so Emma's mitochondrial DNA was thought to have arrived in Northern Europe about 16,000 years before the present. It's called haplogroup K, and it was derived from haplogroup U that was thought to arrive about 40,000 years before the present in South Central Asia. And so the idea is that we can trace back all the populations on Earth back to sort of a common origin. All the current living humans can effectively be traced back to an origin in Africa maybe 200,000 years ago. Yes, that slide is a little bit out of date. I'm going backwards now because Emma's just started work as a Navy dentist. So I told this joke before. I keep saying we should update that slide, but my wife and grandmother and mother-in-law are quite happy with the photos as they are. So Dominic said a little bit about mitochondrial disease genetics. So you've seen that picture of the mitochondrial DNA. There's 37 genes, and nearly all of these have had changes, mutations, which are just changes in the DNA sequence that have been shown to cause disease in humans. and those changes affect at least 1 in 10,000 people, but it turns out that at least 1 in 200 people carry changes in the mitochondrial DNA that could potentially cause disease, and it's maternally inherited. Dominic said a little bit about this mitochondrial respiratory chain or oxidative phosphorylation system or electron transport chain. It's got a bunch of names. As he said, some of the components are encoded by mitochondrial DNA, mostly by the nuclear genes. And so far we know of at least 120 different nuclear genes, and that's why we get to this complexity. Mitochondrial disorders aren't easy to solve because they're not like things that people are more familiar with: cystic fibrosis, thalassemia, Huntington's disease, where there's typically one sort of clinical presentation and there's only one gene involved. So the physician can say, well, if it's cystic fibrosis, we know what the gene's going to be, and in fact it's screened for at birth, and so nearly all children are picked up at birth. With mitochondrial disease, it's not easy like that. It's not a matter of saying, well, we'll just sequence this gene, and when you sequence a gene traditionally, clinically it's going to cost you $1,000, $2,000, $3,000, $5,000, depending on how much it is. There's over 100 different genes, and so you can imagine the costs of having to go through all of those one at a time, at least with the traditional technology. So as Dominic said, the nuclear problems affect probably about the same number of people as mitochondrial, and there's different types of inheritance which he's explained. So traditionally, in children at least, and traditionally in adults as well, we do some DNA testing directly. So some patients are diagnosed directly on a blood test or a urine test for their DNA, particularly for common mitochondrial DNA mutations. But if that doesn't find anything, traditionally it's needed a muscle biopsy, and then we've tended to work out how well is each part of the respiratory chain working and try to use that to guide us to the genetic basis. But it can be very slow and cumbersome, and again, to do those sort of enzyme studies costs about $1,300 for the muscle biopsy, and there's lots of other costs involved in having a muscle biopsy. So it's expensive, cumbersome, and often doesn't give a definitive result. So the encouraging thing is that diagnosis of mitochondrial disorders and other genetic disorders is currently being transformed. It's a really exciting time to be working in this area. And the reason it's being transformed is really an outcome of the Human Genome Project. So back in 2001, the first human genome was sequenced, and at that time, if you wanted to sequence another human genome, it would cost you about $100 million and take months. Obviously $100 million a patient isn't something that any health system is going to deal with. But the cost of sequencing has come down. So this is showing the cost for sequencing our whole DNA, our entire genome, $100 million back in 2001. This is a log scale, so that's a tenfold difference at each of these bars. And up till 2008, it was going down logarithmically, so it had dropped to about 10 million years ago. So 7 years or so ago, it still costs $10 million a genome. And these new technologies have come in so that now in 2013 you can do this for about $10,000 a genome or less. So it's fallen 1,000-fold the cost in the last 6 or 7 years, which is clearly pretty extraordinary, and starts to bring this sort of technology into something that we can start to use. The drop in cost seems to have plateaued out. It may go down again, but it's in the range where we can do this sort of thing. Instead of doing one gene at a time, we can potentially look at all the genes at once, or hundreds or thousands of genes at once. And so the analogy I often use for this is that trying to get a feel for what does 3 billion bases mean, 3 gigabyte bases of DNA. So that's if the Harry Potter series went for 500 volumes instead of 7, then that would be 3 billion letters. It's also, if you looked at War and Peace, it's about 1,000 copies of War and Peace is 3 billion letters. It's a big number, and so that's a lot of copies of Harry Potter. And what are we looking for if we're sequencing all the genes? We're basically looking for a change in one or more letters. Like if you're reading this sentence, if one letter changes, sometimes it's not going to have a whole lot of impact on the meaning of the sentence, and it's just one letter. Sometimes in DNA speak, if you like, you get a bunch of letters that are lost or added in or rearranged, taken from one place and put in somewhere else, and that completely stuffs up the meaning of the sentence. And sometimes you can get very big changes, so maybe deleting all the letters in one volume from the first sentence to the last. So it's a little bit challenging to think about picking up all those different types of changes with one test. but that's what we're trying to do with the new strategies of high throughput sequencing. So to give you a bit of a flavour for this, I'm just going to talk a little bit about a study that we published last year, where rather than sequencing the whole genome, which is still a bit expensive and kind of hard to interpret because it's so big, we've narrowed our focus a little to what we call the Mito exome, where we're basically sequencing a bit over 1,000 genes encoding all the known mitochondrial proteins, and we did that on 44 children with mitochondrial disease, so targeting about 1,000 nuclear genes and the mitochondrial DNA. So instead of 3 billion, that's only 1.5 million letters, so it's a bit of an easier target. It's still challenging. Because when you do that, we're not all the same. If you sequence a whole genome, you get millions of changes between any one individual and the person sitting next to them. And doing this sort of scale, we find about 800 changes in the DNA sequence for each patient we sequence so it's about one change per gene is what we find. And so you have to sort of filter through to find the needle in the haystack, and the way we do that initially is we assume that the sort of changes we're looking for are not going to be common in the population, or everyone would have mitochondrial disease. So we get rid of anything that's present in more than about 0.5% of people in the population, and that takes us from about 800 down to 30 DNA changes per patient. And then we apply some computing strategies to try to work out what is likely to be a problem, what's likely to change the function of a protein, and so that cuts it by about half, ranging from 9 to 33 such changes per patient. And then, we can look at the mitochondrial DNA. If we're looking for what Dominic referred to as autosomal recessive inheritance, where you have to get a badly functioning copy of the gene from mum and from dad, then we're looking for genes that have 2 changes, and so we were typically prioritising 1 gene per patient with 2 changes like that. So that's pretty good. If that works really well, then we're straight away going down to 1 likely gene. And so what did we find in those 44 patients? Well, 12 patients had mutations in known disease genes that were already known about, that we could show pretty easily were clearly causing the disease. 10 patients had mutations in genes that hadn't previously been linked to disease, and we've gone on to prove that they are new disease genes. That's taken about 2 years to get that amount of work, and involved students supported by Amdf and other scholarships. 4 patients where there are changes that we're still working on, that we haven't sorted out whether they're truly causing disease or not, and still almost half the patients where we haven't got any leads yet using this approach, and that's partly because the approach isn't perfect. We keep working on that, and it's improving all the time, but also we don't know all the disease genes yet. And so that study was published last year, and it was done in conjunction with colleagues at the Broad Institute of MIT and Harvard in Boston, and rather nicely was sort of flagged by the US National Institutes of Health as the Genome Advance of the Month for January last year. So it was regarded as a very substantial advance in converting the genetic knowledge into clinical practice, but there's still a little way to go. It's an area that is moving very quickly. The AMDF has supported some work in Perth, trying to use equipment that they had there with good access to do this on patient samples. We are doing these sort of studies partly in conjunction with Boston still, the Broad Institute, and there's also a big initiative in Melbourne at the moment called the Melbourne Clinical Genomics Alliance, involving the 3 Royal hospitals around here, the Royal Melbourne, Royal Children's, Royal Women's, the Peter MacCallum Centre, University of Melbourne, the Murdoch Children's, the Walter Eliza Hall, the Florey, CSIRO, Australian Genome Research Foundation, who've all put in $250,000 each to try to get this into clinical practice. So this technology is moving quickly. It's at a frustrating time when we know we can do it, but to actually get it into clinical practice and get it funded appropriately is still just a challenge. So I think in 3 years' time, I think this will be pretty routine, but we've still got a little bit of way to go to get there. So that's what I was going to say about diagnosis. Diagnosis in this stage where it's truly being transformed, and I think in the very near future, in the next few years, it's going to be vastly improved. So for most patients, it will be doing the DNA test first. Some patients may still need a muscle biopsy to try to confirm what we find in those DNA changes. So in terms of treatment, I think I'll just give the overview and then